Okay, we're going to go over the transcription notes. So we've already went over DNA and DNA replication, so now we're moving on to transcription. All the notes and the extra resources will be in the link below. So this is just a quick little joke, just to remind you of the alphabet of DNA. So DNA, if you remember, has 4 bases: adenine, thymine, cytosine, and guanine. And these are just little baby DNAs saying their ABCs. A, T, C, G. Okay, so we're going over how a protein is made, eventually to the expressed characteristics. So what we're going to be talking about in these next 2 lessons are how DNA goes from DNA and the bases into protein. So we'll be right here. And we're going to be talking about transcription, which is what this lesson is, and then translation, which will be the follow-up lesson. And then we'll eventually be talking about how characteristics are expressed based on the proteins that are coded for by the DNA. Okay, so first we're just going to quick rundown of what transcription and translation is. Transcription is the process of synthesizing a molecule of RNA complementary to a DNA sequence. So that's quite confusing. So in short, it's making a copy of a sequence using the same language of bases. So it's taking that DNA and its bases and copying it exactly. That's what transcription is. Now, translation is the process of synthesizing an amino acid chain whose sequence is determined by the base sequence of an mRNA through the genetic code. So in short, it's converting information from the language of bases to the language of amino acids. So to start out, you have transcription, and you have your DNA bases. So you're copying those DNA bases. You're still in the language of bases, and then when it goes to translation, you're then turning that language of bases into the language of amino acids. And if you remember, amino acids are very important because they are what makes up proteins. So transcription is to make a full copy of something. It happens in the cell's nucleus. So transcription takes place in the cell's nucleus because that is where the DNA is. DNA stays in the nucleus and it doesn't leave the nucleus. That is its home. It's a process, like I mentioned before, synthesizing a molecule of RNA complementary to a DNA sequence. So, RNA is a ribonucleic acid. So this is very similar to DNA, right? DNA is deoxyribonucleic acid. So the big difference is that ribose. So DNA has the deoxyribose sugar, while RNA has the ribose sugar. So there's 3 forms of RNA, unlike DNA where there's only 1 form. So the first form is messenger RNA, or mRNA. It's made in nucleus from the DNA template, and it leaves the nucleus to direct protein synthesis. So unlike the DNA, it can leave the nucleus, and it will leave into the cytoplasm and then to the ribosomes. But we'll get into that a little later. Next, the other 2, we have transfer RNA and ribosomal RNA, which we will also get into when we start talking about translation. mRNA is the key component for transcription, while these 2 are key components for translation. So we're going to go over the differences of DNA and RNA. So first off, DNA is a double helix. Its nucleotide makes up a sugar, which is deoxyribose, the phosphate group, and one of the bases. While RNA is single-stranded, and its nucleotide contains a sugar, ribose, still one of the phosphate groups, and bases. Now, the difference is they have different bases, or one different base. So nucleotide bases are ATGC, and for the DNA nucleotide bases are ATGC, and the RNA nucleotide bases are AUGC. So this thymine gets replaced by this uracil and RNA. Now there's another difference is there's one type of DNA, while there's three types of mRNA, or three types of RNA. So there's mRNA, tRNA, and RNA. So I'll say that again. There are three types of RNA: mRNA, tRNA, and rRNA. So nucleotide-based pair bonding. So there's two main types of bases. There's the purines, always bind with a pyrimidine. So the purines have these two rings, while the pyrimidines have one ring. So the cytosine, thymine, and uracil are the pyrimidines, while the guanine and adenine are the purines. Now this is important because this is how they shape up, right? This is one of the reasons why guanine can't match with adenine, is because, well, then the DNA would be too long, right? It'd be come in real tight and be too long, or too far apart to be more exact. While if the cytosine paired with a thymine, then it'd be too narrow. So this is just a quick of the difference between DNA and RNA. So this is the deoxyribose sugar, and this is the ribose sugar found in RNA. Now the key difference is, see, this is deoxyribose, it's minus an oxygen ribose. So you see you have this OH, and then you have this H. So DNA is missing that oxygen. Now, one of the reasons is elements with a lot of oxygen are more aren't as stable. So by just having one less oxygen, DNA is significantly more stable than RNA, and that's what you want. You don't want your DNA to be able to break down really easily. While RNA, it's not as big of a deal. Okay, so now we're going to talk about the actual process of transcription. So it's making an RNA copy of a DNA sequence, a gene. So we talked about this. A gene is a segment of DNA that codes for a specific protein. So an RNA polymerase binds to DNA. The DNA is unwound into two strands. So remember, DNA is a double helix. So when this RNA polymerase comes in and binds DNA, it splits those two strands. It splits that DNA. So now you have your two strands. Now there's a non-coding strand and the coding strand. Now non-coding strand reads Z to A, while the coding strand reads A to Z. And we'll get more into this later and why this is important. So the new RNA nucleotides added to the non-coding strand to form a single strand of messenger RNA. And the mRNA has the same sequence as DNA coding strand. So this seems kind of backwards because the RNA comes in and it gets its code from the non-coding strand because it wants the sequence of bases for the coding strand. And the non-coding and the coding strand are kind of like mirrors of each other, right? So it makes sense that if it wants this code, it would need its reflection because it'd be reflected back to get that coding strand. If that seems confusing, don't worry, because we're gonna have a lot of practice with this. So this is non-coding, or is also known as a template strand, and this is the RNA transcript or mRNA right here, and this is the coding strand. And then here's the mRNA when it leaves the nucleus. So this is what happens. So the mRNA polymerase comes in, it splits the DNA, and if you can tell kind of this picture, this is the double helix, this is the double helix, so it splits the RNA, comes in, it starts laying down these bases, grabbing the bases for the complementary pair. So you got A, U, see this is that uracil that replaces that thymine, right? Because usually adenine pairs with thymine, but in the case of RNA, it's adenine pairs with uracil. So now you have RNA transcript and RNA. Now when that's done copying, it'll leave, the DNA will close back up to its double helix. It'll leave the nucleus and enter the cytoplasm. Now, if you can tell, this right here, this strand, which is this mRNA that was copied from the non-coding strand up here, is the same as the coding strand, right? You got the U, T, A, C, U, T, and uracil and thymine are very, very similar. So that's why they're considered the same in that case. And we'll get more in the why the differences and the similarities between uracil and thymine later on in this unit. So just for clarification, because there are some books out there, because terms and science terms change over the course of time. So some of our books or some of the papers or worksheets you'll get will say sense strand and anti-sense strand. Now, just important to know that coding is equal to sense strand and the non-coding is equal to anti-sense. Just all you need to know there. So just some quick practice. So we have our non-coding strand right here, which is a AGT, Now this is just random bases I picked out. They don't necessarily represent anything. See, now here we have our mRNA, messenger RNA. See, you got the uracil, cytosine, adenine, cytosine, adenine. See how this matches? See, although, but yeah, see this matches GG. You got the T to the A to U, CG, AU, CG, AU. So that's it for transcription. That was very fast. If you have any questions, leave a comment or talk to me in class. The resources, so the notes, there will also be a video posted that will go over transcription and translation that we watch part of it during this lecture, and we'll also watch part of it during the translation lecture, and also provide an additional video that talks about transcription. Thanks for your time.